We will be investigating the process of EJC recognition-dependent NMD, or Exon Junction Complex recognition-dependent nonsense-mediated decay. As mentioned from Dr. Green's lecture, nonsense mutations contribute to an array of diseases including cystic fibrosis. Ribosomes conduct mRNA and protein surveillance in order to detect premature stop codons, or premature termination codons known as PTCs, located on mRNA molecules. After detection of PTCs, ribosomes can then target the mRNA molecules for a variety of pathways, including nonsense-mediated decay, referred to as NMD, no-go decay, and non-stop decay, as stated by the 2018 review article entitled Lost in Translation, Ribosome-Associated mRNA and Protein Quality Controls by Karamyshev and Karamysheva. However, we will focus exclusively on NMD to remain consistent with Dr. Green's lecture. Truncated polypeptides produced at the stalled ribosomes are ubiquitinated and degraded by the proteasome. The regulation of aberrant protein production pathway, also known as the RAPP pathway, senses aberrant proteins. It does so by scanning the newly assembled protein side chain interactions during translation and triggers degradation of their mRNAs. If this step is missed, the truncated polypeptides are degraded by the proteolytic machinery in the cytosol or endoplasmic reticulum by ERAD. If this step is missed, these mutated proteins will form insoluble aggregates known as amyloids and result in disease. The Exon Junction Complex, or EJC, is a complex of proteins usually assemble 20 to 24 nucleotides upstream of exon-exon junctions during pre-mRNA splicing. After mRNA export from the nucleus, EJCs are removed during the pioneer round of transport and replaced with proteins that promote translation. The final normal termination codon is usually located in the final exon and, thus, does not have a downstream EJC complex that needs to be removed. The general rules regarding the distance between PTCs and EJCs regarding nonsense-mediated decay are summarized as follows, according to the 2016 article by Pop and Makwat entitled Leveraging Rules of Nonsense-Mediated mRNA Decay for Genome Engineering and Personalized Medicine. If the PTC is located greater than 50 to 55 nucleotides upstream of an EJC, NMD is activated. The terminating ribosome at the PTC during the pioneer round of mRNA transport and development is sufficiently far enough from the EJC such that the terminating ribosome is not able to remove the EJC. Because of this, the presence of the EJC is able to initiate a signal cascade, as will be touched on later in the video. If the PTC is located less than 50 to 55 nucleotides upstream of an EJC, NMD is not activated. The terminating ribosome at the PTC during the pioneer round of mRNA transport and development is sufficiently close enough to the EJC such that the terminating ribosome can remove the EJC. Nonsense-mediated decay will not occur for this PTC because there is not an EJC present to initiate NMD. There are many important proteins for the EJC recognition-dependent NMD pathway that will be explored here. The first category is up frameshift proteins, UPF1, UPF2, and UPF3. UPF1 is known as a master regulator of nonsense-mediated decay from the 2018 Karamyshev and Karamysheva article. The ATPase activity of UPF1 is necessary for the disassembly of mRNPs during NMD. UPF3 can exist in two forms, UPF3A and UPF3BX, in mammals that will be explored in the signaling pathway of NMD. Other proteins that help to regulate NMD include the following. SMG1 is a phosphoinositide 3-kinase P1-3K, like enzyme that phosphorylates UPF1 at multiple glutamine sites. SMG 5 through 9 all help regulate NMD, as will be explored in the signaling pathway of NMD. The 2011 snapshot Nonsense-Mediated mRNA Decay by Durand and Anderson, and the 2018 Karamyshev and Karamysheva article were used as the basis to understand the following pathway. The first stage, stage 1, involves detection of nonsense-mediated decay substrates. This occurs during the pioneering round of translation and involves encountering a PTC greater than 50 to 55 nucleotides upstream of an EJC. UPF3A or UPF3BX binds to the EJC. Stage 2 involves tagging substrates for nonsense-mediated decay. This tagging involves formation of the SIRF complex at the terminating ribosome. The SIRF complex includes the following: serine or threonine kinase SMG1, up frameshift protein 1, and eukaryotic release factors ERF1 through ERF3. Stage 3 involves degradation of the mRNA containing the PTC. The UPF2 binds to the UPF3A or UPF3BX. Next, the UPF1 SMG1 complex interacts with the UPF2, which brings this close to the EJC. Once interacted with UPF2, SMG1 phosphorylates UPF1. Hyperphosphorylated UPF1 causes translational repression and serves as a recruitment platform for other SMG proteins. Specifically, SMG6 is recruited, which performs endonucleolytic cleavage of the mRNA between the PTC and EJC sites during the last stage of NMD. SMG 5 through 7, or SMG 5-PNR-C2, are also recruited, which recruit decapping and deadenylation machinery. These facilitate exonucleolytic degradation of the unprotected 5' and 3' mRNA fragments following endonucleolytic cleavage by SMG6. Further degradation proceeds as follows. XRN1 is a 5' to 3' exonuclease responsible for the degradation of the 3' cleavage products. The exosome is responsible for the degradation of the 5' cleavage product. In summary, nonsense mutations contribute to an array of diseases, and ribosomes conduct protein surveillance to detect PTCs. PTCs are detected and targeted for nonsense-mediated decay if they are greater than 50 to 55 nucleotides upstream of EJCs. Many proteins are involved in the signaling pathway for EJC recognition-dependent NMD, including upframe shift proteins and SMG proteins.